I think we will go ahead and get started. We're really excited that you are all here today. This is a follow-up to a webinar that we taught a couple of months ago, talking about Western Bumblebee ID, and we ran out of time to talk about males at the end. So we decided to have a separate webinar to just talk about male Bumblebee ID. So hopefully you're in the right place. We're a small but mighty crowd. This is definitely an esoteric subject to a certain degree, but we're excited to dive in and share some of the knowledge that we have. My name is Rich Hatfield. I'm a senior conservation biologist for the Xerces Society. I'm based in Portland, Oregon. I direct our Bumblebee conservation programs, including running the Pacific Northwest Bumble Bee Atlas, and I also sort of oversee a lot of our other Atlas projects, which will be in 20 states next year. So we're growing again. So anyway, Leaf's going to start us off with a sort of an intro, and you'll see me again a bit later, but thanks for being here. And yeah, let's get started.
Hi, everyone. I'm Leif Richardson. I'm a conservation biologist with the Xerces Society for Invertebrate Conservation. I live in Southern California, and I lead the California Bumble Bee Atlas. So Rich and I work closely on these Bumble Bee Atlas projects with our other colleagues scattered around the US. So I'll start just by saying a few things about Zoom. Please, if you have questions, type them into the Q&A. And while I'm speaking, Rich may type a few answers or flag a few of those questions for answering live, and I'll do the same for him. Please try not to put the questions in the chat. It'll just make it cleaner for us to find them in one place. And we will have a little question and answer at the end of the session. We have 40 slides, and so we're hoping to bring this in at about 40 minutes, so we have plenty of time to talk. So yeah, let's get started. I'm going to share my screen. Rich, can you just give me a thumbs up if that looks good?
It looks great.
All right, okay. Good. Okay, so welcome to Identifying Male Bumblebees in Western North America. This is, as Rich says, a little bit esoteric. We mainly teach female bumblebee identification. And then we, most of us who are so-called experts, we started with the females and maybe focused on them for a long time. Males are, if you think the females are hard, I think the males can be even harder. And so we typically don't try to put this in front of Atlas volunteers right away, but lots of you are very avid about ID, and this is completely in your wheelhouse. So we want to present this tonight, and hopefully it's useful to you, and we're happy to follow up with discussions or conversations about the material. First, a few things about the Xerces Society for Invertebrate Conservation. So we protect wildlife through the conservation of invertebrates and their habitat. We definitely think of invertebrates as wildlife, but of course, they are important to all sorts of wildlife with and without backbones. We have the following programs that we organize our work around: one about native pollinators, one focused on endangered species, which is where Rich and I work, one on aquatic invertebrates, for example, mussels, butterfly conservation, where we work on monarch butterflies quite a bit, but we also are working on all of the other native butterflies in the West. And then a program on reducing pesticide exposure for invertebrates. We are an evidence-based organization. We use science to inform our decision-making. We work with lots of partners in academia, in government, private researchers, of course, community scientists, like many of you are. And again, we take that information that we glean from the field and use that to make decisions about how to invest resources in conserving and managing invertebrate populations. Community science, also known as citizen science, is a really important part of what we do at Xerces. Rich and I are heavily involved in these Bumblebee Atlases. It says 15 states here, but as he said, there will be 20 states active next year. We have other community science projects as well. Bumblebee Watch is the platform for the Bumblebee data, but we have several different projects focused on monarch butterflies. So we counting Western monarch butterflies in their hibernating areas in California on the West Coast. We have one mapping the host plants for the monarchs, the milkweeds. We have a freshwater mussel survey, a new firefly atlas, and more. So there's a link at the bottom there if you're interested in getting involved in other community science projects. And yes, those Bumble Bee Atlas projects are examples, and here's the map of the states where we will be active next year. Montana is another state where we will be active. So with Montana, the green states are 20 states, and we have many, many partners who make this work possible, and some of them are indicated to the right there. Our work, we're a nonprofit, and our work is made possible by membership and donations. So if you're a member, thank you so much for supporting our work. If you're not and you'd like to be, there's the link for learning how to do that. Thank you. All right, let's get into bumblebee identification. So how do we go about identifying things? Well, in the invertebrate world, in the insect world, scientists who are specialists mainly use dichotomous keys, like the text you see on this slide, which is copy-pasted from a bumblebee key for bumblebees in California. And so by dichotomous, I mean there are two choices at each sort of question. The first one at number 1, it says, do you have a female bee or do you have a male bee? And so you have to make a choice, and they're mutually exclusive in most cases. And so let's imagine we have a male bee, and so it goes to number 25. So we go down the key to number 25, and then we read those two possibilities and think about which one is more realistic for the bee that we have in hand, and move forward until we get to a species. But at Xerces with the Bumblebee Atlases, we don't destructively sample bees, we don't kill bees, and we don't use, therefore we don't use microscopes in the lab to try to identify bees using these very complicated keys like this. Instead, we are doing photo-based ID, right? I think virtually all of you are familiar with this. So it's a little bit different. There are certain characters we can't see with photos only, and we do need high quality photos from a number of angles to identify the bees. And we are focused on hair color. And that is despite the fact that hair color is not necessarily reliable as a way of identifying bumblebees. It's simply the best thing that we have for identification based on photos. We will also use other things like morphology, but as this bullet here says, there's a sort of famous bee scientist who described the bumblebees as morphologically monotonous, meaning if you shaved all the hair off of them, they all look pretty similar, and it's hard to find sculpting on the exoskeleton or other sorts of characters that can help you with this. So we are going to teach you to use the hair color quite a bit here. But we're going to remind you frequently that there are some character states that we just cannot see in photos and that we need in some cases with male bumblebees. And here they are. These are the main ones. These are the genitalia of male bumblebees extracted from the bees, the photos on the right. So if you're collecting bees and killing them for ID, you tweezers to reach up inside the back end of the abdomen and gently pull the genital capsule outward, and that includes these male parts. And the specific little structures that you can see there are used in the keys to distinguish the various species. That is not something we teach, and it's not something we do, Rich and I, very often at all, but we just want you to know that is necessary in some cases for identifying bumblebees. And so in some cases, we just can't identify them with our methods. Let's talk a little bit about morphology and distinguish males from the females. So both have 2 pairs of wings, so 4 wings. They have 4 wings and hind wings. Sorry, that's confusing. The males do not have a pollen basket or orbicula or scopa. Those are all synonyms for that thing that the females use for carrying the pollen. The males simply lack it because they do not gather pollen for the nest. Males have 1 additional abdominal and sternal segment. Sorry, tergal abdominal segment and sternal abdominal segment. So that's tergal are the top ones, and sternal are the underside or chest side of the bee. So they have 7 of those, and all 7 are important in ID. And we teach you females. We do not talk about the 7th one, so you'll hear a little bit about that tonight. The head is similar for both males and females, except one important difference, which is that males have 13 antennal segments. Well, females have just 12. And so these 3 things, the presence or absence of the scopa, the number of abdominal segments, and the antenna, the number of antennal segments, those are the things that we use to distinguish males from females. Once you get good at it, you can tell without necessarily seeing those things in most cases, but Those are the three main character states that you could use if you're a beginner to distinguish the males from the females. I'm going to show a few other slides with morphology, and my apologies, some of these feature female bees. Just for simplicity, I grabbed this from one of our other talks, but this is the face or the head of the bee. I should say the head of the bee. The face is the area indicated right in the middle between the bases of the antenna. The vertex is the top of the head, right up top there where it says vertex. The ocelli are those simple eyes at the vertex, whereas the compound eyes are the big ones that we're familiar with. We're showing you the antennal segments here. We're not going to get into talking to you about A6 versus A7 tonight, but if you dive in deeply to male bumblebee identification, you're going to find that you sometimes are asked to compare the lengths of different antennal segments. And so that's why we start to teach this. Note in red down there, A13 or F11 is missing in this photo. Obviously, it's a female and not a male bee. Here is the face of three females from head on. And here we're illustrating the difference between a long, a medium, and a short cheek length from left to right there. And the brackets are showing you that. And so by long, or the dimension is from the base of the compound eye to where the mandible or mouth part attaches to the head. And that's indicated by the bracket there. Ultimately, we're really interested in the length versus the width. But sometimes people just express it as length here. And so with males, this is also important, although it's a little bit less often used when identifying the males. So as with the females, and these are females indicated in these little cartoons, as with the females, the males form these complexes of very similar looking species in one place. We call them mimetic complexes, and the thinking is that they have evolved to look similar to each other in habitats that they share, so that predators will be more likely to avoid any bee that looks like the one that is dangerous to them. There's lots more to say about that, but just the only reason I'm bringing this slide to your attention is that one of the reasons it's so hard to identify male bumblebees is that they can look frustratingly similar in any given place. And that's similar to what we've talked previously about the females. So get ready for some challenges here. We're going to organize the rest of this talk around this flow chart or the character states on this flow chart. So this is a very simplistic way to identify male bumblebees from photos. And the basic idea here is to find some easy to identify morphological or hair color pattern character states that only a few of the species have, and then move from left to right with successively more difficult characters or characters that are shared by more species. I have the species names at the bottom here, and they're not exclusive groups on the right side. So there are some. So if we identify a bee with big eyes at step one, that bee may also have T1 yellow on the way to the right, but we're not listing it down there because it's an iterative process starting on the left. So big eyes, you'll hear more about this, but there's a photo of a large-eyed bee there, much larger than an average bee. If your bee has big eyes, you would then consider one of these 5 species below. If not, you proceed to the question, does it have a red tail? Is there red hair on the tail? Like in the next photo, and there are 4 possibilities for you there in male bumblebees. What about if it's got light colored hair, but it's really not red, it's sort of yellowy, dingy, off-white, pale. There are 3 species there. If it's not like that, we next move on to bees that are mostly yellow, and there are 4 possibilities there that you'll hear more about. If not that character, then we're moving on to a taxonomic group, which is the cuckoo bumblebees, and we'll teach you a little bit about how to identify a bee as a cuckoo bumblebee a little bit later. Finally, we get into the larger bins here. Is the first abdominal segment, T1, is it black? And you see the bees below. And then finally, if none of those other character states apply, is T1 yellow? And that's all the rest of the species. I should say this is 27 species. In the West, if you included Northern Canada and Alaska, there's probably more, like 40 species, but we're mostly sticking to the United States and adjacent Western Canada, so 27 species here. And the ones in parentheses appear a second time there because they have distinctly different color patterns in different places, so we wanted to include them. So let's go through those categories. Here are some big eyes. There are two male bees on the left side with large eyes. At the top, that's called Bombus nevadensis, and at the bottom, it's Bombus crotchii, so Nevada bumblebee and Crotch's bumblebee. If you look at the eyes, they are much larger than the average female bumblebee's eyes. And average, you will have to learn what's average by experience. The bee on the right is a female of Vosnesensky bumblebee, and she has those average eyes. They're not too big, they're not too small. That's an average looking bee. But just look back over to the nevadensis, huge eyes. They are just gigantic. The bees in the middle here are examples of females of crotchii, and they have relatively normal sized eyes also. So big eyes, they're usually pretty easy to spot, with one exception, which we'll talk about a little bit later, Bombus rufus cinctus, which has big eyes. Moving on to the bees with red tails. This is pretty self-explanatory, but it's red hair found on T4, 5, 6, and or 7. Well, at the end of the, it's usually T7 and back as far as T4, I should say. It can be bright red. It can be a really sort of beautiful orange. It can be a light orange. And in some specimens, unfortunately, this hair is really washed out and is almost yellowish, which will make it confusing that I've divided us into red tails and pale tails. So just be aware that that red can fade or can present as a lot lighter, and rarely in some specimens. And that's Bombus mixtus there. Next is the pale tails. I have two of the three species here. On the left, we see Sitka bumblebee, or Bombus sitkensis, and you can see a lot of yellow hair on this guy, but especially look at the tail there where the arrow is pointing. It's distinctly white or off-white, and this is the same individual bee, and you can kind of see from the different angles that indeed he has a tuft of white hair there. On the right, you see the western bumblebee, or Bombus occidentalis, and that bee usually has a very prominent stripe of white at the end of the tail, 1 to 3 abdominal segments. I'm working from memory there. Unfortunately, in coastal California, this western bumblebee, the white hairs can appear instead to be kind of yellow-orange, which makes it hard to call them pale. And some of these other species can rarely have a little bit darker hair. So the red tails and the pale tails, personally, I find them pretty easy to distinguish, but that's with experience. So you're going to have a little humility while distinguishing those two groups until you get good at it. The next group is kind of fun. It's bees that are mostly yellow. They have a ton of yellow hair, usually on the face. The thorax is usually almost all yellow, and then largely yellow on the abdomen with some other colored hair in there also. So here's Bombus flavorfrons in that category. And then the cuckoo bumblebees. As I said, this is a taxonomic group where we're not showing you one single trait to look for, like pale hair. But I've got the 3 species here that occur in most of the Western US, and in Northern Washington, you could possibly get another one. But anyway, you'll hear more about these bees from Rich in a minute. But the 2 characters I would recommend looking for to identify male cuckoo bumblebees is the first one on the left, indicated by the blue arrow. It's got a kind of a thick head. Behind the compound eye, you can see a shiny area that is integument. It's the backside of the head. And that area is just a bit thicker than you see on bumblebees that are not cuckoo bumblebees. This is a subgenus of cuckoo bumblebees. And then the other one is the tarsus of the hind leg, where female bumblebees that are not cuckoo bumblebees carry pollen, right? Males obviously don't carry pollen, and so they don't have that pollen basket. The cuckoo males have even less of a pollen basket. I guess they have somewhat of a convex shape to that body part. It's usually not at all shiny, and it is usually stippled with hair all over, which I think you can see in this photo, and that distinguishes them from some of the other males. Moving on to T1 being black. This is the first abdominal segment. It's the one that faces the thorax, and it can be difficult to see in photos. Like this photo, I think you can just barely make it out under the wing and above the leg there. Sometimes most of T1 is kind of vertically facing the thorax when the abdomen is tilted up, and it's easy to see to miss it and then count T2 as T1. So try to prove to yourself that you can count segments on a male bumblebee. For reference, the yellow stripe on this bee is T4. And this is this is actually, I don't know. This is either Voznesensky bumblebee or Calagen, fogbelt bumblebee, and I cannot distinguish them from photos. Finally, we have bees where T1 is yellow. And so this is Bombus vagans. This is a beautiful species that's found in the east, quite broadly distributed, and then works its way north and west. It's found in Washington and Idaho and possibly Oregon and farther north. So they have 2 stripes of yellow on the abdomen, including the first segment. It's usually clear, just all yellow in the species that have this situation, but sometimes you'll find some black hairs, often on the sides of the segment, like right where the arrow is pointing. That's the lateral side of that T1 segment. Again, let me just remind you that that we're going left to right in this little heuristic. And so some of the bees that have big eyes have T1 black also. So we're ruling them out at big eyes. So when you get to T1 black, it's just the remaining species. Okay, so that can be somewhat confusing, but I hope you'll see that this is a little bit like a dichotomous key where you're choosing between 2 possible character states. And with that, I'm going to turn it over to Rich. I think I'm going to unshare my screen and he's going to share his. 1 second.
How are we doing, Leaf? Am I showing my screen?
Looks good.
Okay. Can you see the Zoom bar on there or no?
Sorry, can I see what?
The Zoom menu bar is over that screen, and I just want to make sure it wasn't obscuring anything.
I think it's okay.
Okay, great. Okay, actually, while we transition here, there was a good question that popped up. It was a question about corbicula, and the question is, I answered it in the chat, but I think it's a good question that a lot of people maybe haven't seen or maybe heard the answer to. Are scopa and corbicula used interchangeably? If not, what are the bees in addition to bumblebees with corbicula?
Yeah, so in Rich's answer, I'm reading it. A corbicula is a specialized scopa, and honeybees and bumblebees have a corbicula. And basically, it is a shiny, hairless, concave area on that hind leg tarsal segment that has bristly hairs that arch over it. That's to be distinguished from other types of scopa. For example, a really common way that they look is just a brush of hairs, and the bee is packing the pollen, dry pollen, up into the brush and using electrostatical energy to keep the pollen there. But these bees, honeybees and bumblebees, they moisten it with nectar and make a kind of bee bread that they put there. And Rich is correct. Orchid bees, stingless bees, and a couple of other types are true corbiculate bees.
Great. Thanks. I'm gonna take things over from here and start walking through, at least introduce these sort of broad categories, kind of like we do with the females, where we have, the white tails and the yellow tails and the different groupings that we've presented to you. I think it's what we present to you with females, I think it's easier to categorize those animals into the groups in which they belong. I think there's a fair bit of crossover here. So while Leaf says when you get to T1 black, in theory, you should have eliminated the big eyes, the red tails, the pale tails, the mostly yellows, and the cuckoos, you may have made a mistake. And so if you get confused or don't think you may not have the right species, I think you just have to remember that this is not a dichotomous key. It's not a perfect tool. It's just it's a kind of like our female guide. It's an entry point to then allow you to pick up the Bumblebees in North America guidebook or another guidebook to try to finalize a name. And that's really the goal of what we're trying to present for you here. So anyway, what I'm going to do now is kind of look at some of these categories like big eyes and look at some of the species within them that could be confusing and give you a few characters that Leaf and or I use to distinguish these species when we are looking at photographs. So we're going to start and sort of follow through here, beginning with the big eyes and moving to the red tails. Actually, I don't think I actually have any red tails that are confusing, but we're going to move basically from left to right through this flow chart. And we are going to start with Nevadensis and Morrisoni, and then we're going to talk about the yellow teddy bears, and we're going to talk about the fuzzy wuzzy yellow teddy bears, and the wasn't fuzzy yellow teddy bears, the ones that have longer hairs than others. Then we'll talk about some cuckoo bees, those with T1 black, some with T1 yellow, and then we'll talk about Rufosinctus, which of course, as the females are sort of in a category of their own. So jumping in, we will talk about distinguishing Nevadensis from Morrisoni. If you remember from the females of these two species are also very difficult to distinguish from one another. They live in the same region. They're sort of intermountain west species. So shrub steppe, kind of drier habitats. They have a mostly yellow thorax, and they have the first three segments of their abdomen that are yellow. One of the key things that we look at with the females is that the Nevadensis females have a black dot in the middle of their thorax, whereas Morrisoni are completely yellow in that area. That character still holds slightly with this, but female or male Nevadensis can be quite a bit more yellow. I will say that the character of the hair colors that you see on this slide here are maybe not diagnostic, but they're a field character you can use. Morrisoni does tend to be this really beautiful golden yellow color, whereas Nevadensis tends to be more straw-like in color. But one of the things that we look at in particular with these is, in addition to having big eyes, which I'll show you a closer picture of their big eyes in a second, so these are going to be in that big eye group, is actually looking at the tail. So you'll notice if you look at Bombus Nevadensis over here on the left, that I'm circling an area right now that appears red. And that's not a light aberration or anything. Those are actually red hairs that are on the tail on T7 of Bombus Nevadensis. And Bombus Morrisoni lacks those red hairs. So we don't have any red hairs over here on the right. So that's an area that you can really look at to try to distinguish these two species from each other. Sometimes you will get a dark Nevadensis that doesn't have those red hairs, of course. So there's other things that we can look at too. And for that, we're going to look at the eyes. And Leaf showed you this picture earlier of Bombus Nevadensis on the left, where you can just see those giant eyes. And similarly on the right, and I'm sorry this photo is not perfectly in focus, but it was the best photo that I could find for this particular purpose. They are also quite large. But one thing that I'd really like you to notice is that near the tops of those eyes, those large eyes, on Bombus Nevadensis, they converge. The tops of the eyes are closer than the bottoms of the eyes. And that is not true with Bombus Morrisoni, where those eyes are more parallel in nature. So they're not converging near the top. And that is one of the key things that you might look at to try to clarify whether you have a Bombus Morrisoni or a Bombus Nevadensis. And in fact, this is a character you can see in the field. You don't need a microscope to do it necessarily. But this is one of the key, this is one of the items in the dichotomous keys in the back of the book to distinguish these two subgenera from each other, is actually looking at whether the eyes are highly convergent near the top or not. So this is a good character to look at to separate Bombus Nevadensis from Bombus Morrisoni. Next, we're going to move on to these bees that were mostly yellow. We familiarly call them the yellow teddy bears because they're well, they're teddy bear-like and that they kind of look cute and fuzzy. Some of them are more fuzzy than others. So I'm going to sort of divide these up into the ones that I think are sort of less fuzzy, and then the ones that are more fuzzy. And I think you'll see that in these photographs. So the first group is the wasn't fuzzy group, the ones that aren't quite as fuzzy, where the hairs are more even in length. They're not sort of long and shaggy. They tend to be more even in length. And there's 3 species in this group. Those 3 species are Bombus fervidus on the left, Bombus appositus in the middle, and Bombus pennsylvanicus on the right. And generally speaking, they all look fairly similar to each other in that they have a black band on the thorax, and then most of the abdominal segments are yellow. To distinguish these 3 species from each other, we're really going to be looking at the tail. If you look at Bombus fervidus, which is the one over here on the left, you'll notice on the final abdominal segment, actually I think this is T6, there is yellow in the center of T6, but the side of T6 here is black. Okay, and this is a character that we often, not always, but we often see in Bombus fervidus. The opposite is true for Bombus appositus, where on the side of that same segment, you see yellow hairs, but in the middle of that segment, you see black hairs. And then over here on the other side, you would also see yellow hairs. So if you can see my annotations here, for Bombus appositus on that abdominal segment, you're going to see yellow on the sides and black in the middle, and the opposite for Bombus fervidus. For Bombus pennsylvanicus, it's usually going to have a black segment there, but behind it on T7, just like we saw in the photo I showed you earlier with Bombus nevadensis, Bombus pennsylvanicus usually, and so I use this term liberally because it is not always, but this species usually has red on T7. So this is a good character to look for to separate out these 3 species. Generally speaking, Bombus appositus is also going to have a little bit more yellow on the face than either of these other 2 bees, but that character in my experience is quite variable, and you're mostly going to want to be looking at the tail of the bees in these last couple of abdominal segments. Leif, I should say at any point, if you want to jump in and say something I'm forgetting or not, please go ahead and do that. And you can see these things in the keys as well. On Bombus fervidus, you'll see on T6 here, there's yellow in the middle. And then on Bombus appositus, you see yellow on the sides. And for most color patterns for Bombus pennsylvanicus, especially for the ones that we'd see in the West, which are the Bombus sonoris versions here on the left, they have yellow on them, or red on the very end of the tail. Sorry if I cut you off there, Leif. And then here are the fuzzy yellow teddy bears, 2 particular species that we wanted to look at because both Leif and I struggle with identifying them, especially from photographs. And as you can see, these 2 species do look quite similar. But what we're really going to be looking at for these 2 species to distinguish them from each other is starting on the 3rd tergal segment. So here's the end of T1. Here's the end of T2, and this is the start of T3 here. Over here, the end of T1 is here, the end of T2 is here, and this is the start of T3. So we're looking at T3. And if you notice, well, it's a little hard to tell, but for the most part, if I look at the hairs in this region that I'm circling here, they're all yellow. There are no black hairs in that region of it. It does look like there's sort of an aberrant colored hair right there. But I really think that's more we're seeing through the hair and actually seeing the integument of the bee, whereas those actual hairs are yellow. In contrast, if we look over here in this region by the arrow over here, you can see that there are some black hairs that are appearing at the front of T3 and at the sides, and then even more on T4. We can start to see more black hairs down there on T4. And so this is a character that we use to distinguish these two species from each other. With yellow hairs only and no black hairs, that's Bombus van Dijkii on the left. With black hairs, we have Bombus flavifrons on the right. This is a difficult character. It takes a high quality photograph in order to be able to see it with certainty. There are a couple of other characters we can look at as well. Bombus flavifrons has a longer cheek than Bombus van Dijkii, but for the most part, this is the character that we are looking at with these two species. And their ranges are somewhat similar as well, so not a great distinguishing factor either, except when we get much further east than most of the people on the webinar here are today. Then Bombus van Dijkii drops out of the picture. And again, you can see this on the keys. So here's Bombus flavifrons. You can see that those black hairs show up as sort of being gray at the front of T3, although you can see if you look at some of the color patterns, that isn't always true, or what is really is it's less true. And that's why I said really pay attention to the sides. Look at the very sides of the abdomen, and there will be almost always a couple of black hairs there, which will distinguish Bombus flavifrons from Bombus van Dijkii, which has no black hairs in that region. Obviously, sometimes Bombus flavifrons can also have red on it, and that would help distinguish it quite easily from the species, but that's not what we're focusing on in this slide. Here's just another example of Bombus van Dijkii versus flavifrons. Here we have flavifrons on the left. You can see those black hairs intermixed there, and then here on the right, there are no black hairs intermixed on the side. Those are all yellow hairs, so we can tell that it is Bombus van Dijkii and not Bombus flavifrons. Next, we're going to move on to those cuckoo bumblebees. We've sort of moved through a couple of stages in that flow chart, and now we're looking at the cuckoo bees. On the left here, you have a cuckoo bee, and on the right, you have a true bumblebee. I actually think this is Bombus flavifrons here on the right. You can see the somewhat large long cheek here. But instead of looking at the cheek in the situation, and Leif alluded to this earlier, we're actually going to be looking at the head itself. The cuckoo bumblebees, especially the males, are what we call the big-headed bumblebees. And I've tried to draw some lines here to help just show you a little bit. We're looking at this space behind the eye. So the eye of the bumblebee, or this bumblebee, is here, and the eye of this bumblebee is here. These are both regular sized eye males, so they're not bulging eyes. But if you look at the space behind the eye, but in front of the thorax, you can see that there's this big bulbous, sort of shiny space behind the eye in the cuckoo males. And that's something that we see. And I also drew this horizontal line here because this sort of hat space that you see up here, this kind of tall head, it's actually above the eye. If you were looking from the front of the bee, you would actually see like a little bit of space above the bee. That's something that you can look at to try to distinguish the cuckoo bumblebees from the true bumblebees. So again, true bumblebee here on the right, where there's less space behind the eye and a lot of space behind the eye in the cuckoo bumblebees. Realize these are all relative terms, so it takes seeing them a lot of time, but these are things that we're looking at when we're trying to distinguish what species of bees that we're looking at. Once you've decided that it's a cuckoo, there's a couple things that you can look at to try to distinguish the two most common cuckoo bumblebees that we have. That's Bombus insularis and Bombus flavidus. Insularis is on the left here, and Bombus flavidus is on the right. What we're really going to be looking at is again towards the tail of the bee. Bombus flavidus is going to have a solid, for the most part, solid yellow band on T4. And Bombus insularis is going to have black centrally on that same segment. So we're going to be looking for that. Additionally, Bombus flavidus is one of those red-tailed bees. So it's going to have, almost always, in my experience, it's going to have some red hairs at the very tip of its abdomen. And those are the things that we're going to be looking at. The rest of the body are quite variable and very similar, so hard to look at. are hard to distinguish. So really, again, looking towards the tail of the bee and the color patterns towards the tail. Those are the features that we're looking for. Next, we're going to move on to the T1 black bees. So we will have eliminated a bunch of other bees, but now we're on to these species where the first abdominal segment is black or mostly black. The two species that you see here, I don't know what they are. I have no idea. One of them, I think the one on the left, if you were to sort of hold my hand to the fire and make me put a species name on it, I would tell you that the one on the left was Bombus vazasensky. And if you put my hands to the fire on the right, I would probably tell you that I thought it was Bombus caliginosus. But I'm really just guessing here. I'm going mostly on the shagginess of the hairs, particularly on the abdomen. It's really not shaggy on this bee on the left, and it's quite shaggy on this bee on the right. But males are males, and they're fuzzy, and they're wuzzy, and they're confusing. But otherwise, these two bees have identical color patterns. For the females, you'll remember between these species, we use the sternal segments. That goes out the window with the males because they're yellow on both species. So we can't use that to distinguish between these. The cheeks are somewhat similar in length. They're slightly longer in Bombus caliginosus, which is one of the reasons why I think this one on the right might be Bombus caliginosus. Its cheek is slightly longer, I think, than this one on the left. But again, I don't have the perfect angle. I can't measure it under a scope. So we can't really distinguish these two. If you've submitted photos of males, yellow-faced, yellow-striped bees, most likely you've gotten a verification of Bombus caliginosus or vazasensky back, unless it's significantly inland and Bombus caliginosus is just, we believe, virtually impossible based on locality, because the fog belt bumblebee Bombus caliginosus is really only closely associated with the coast and the coast range along the Pacific coast, whereas Bombus vazasensky actually makes it all the way to Idaho. So we really can't tell the difference between these two species from color patterns. In order to do that, we would have to, as Leif mentioned, we would actually have to pull the penis capsule out of the bee and look at the penis valves. And again, we're not going to do a lot of this tonight. So don't turn off your webinar because you don't want to talk about penis valves. This is the only slide that we have besides the one that Leif's already shown you. And you can see there's some differences in the structure of these. I've drawn some lines here to hopefully help you see that. For Bombus caliginosus here on the left, its penis valve is a little bit thicker and less pointy. And then Bombus vazasensky here on the right, its penis valve almost comes to a point at the end. So the shapes of those two are different enough that we can see them through a compound microscope. Not something that you can see from a photograph, but if you have a bee on a pin, this is how taxonomists distinguish between these two species. Again, this takes a lot of experience, and if you can only see one of them and you don't have the reference material in front of you to compare it, it gets very, very, very challenging to make these determinations based on that. So I gave you a grimacing emoji for this one because I think this is a hard choice to make, even if you have the taxonomy in front of you and the specimen in front of you. These are two very difficult bees when they're males to distinguish from each other. More T1 black bees, Bombus fervidus on the left, Bombus flavidus in the middle, and Bombus insularis on the right. So this is one of those times, right, where there's some crossover between the species. In theory, you would have already eliminated Bombus flavidus and Bombus insularis because we've been past the cuckoo bumblebees. But my guess is that some of you might miss that, right? And so, because it's not always easy, you might not have a good view of the back of the head, and it might be, you might end up with them in the T1 black category. So I'm just going to go over sort of some differences between these three species because of that. For the most part, these species are not going to have those bright yellow hairs that on their face that we just saw in caliginosus and vazasensky. There's a significant amount of black mixed in on all of them. So their faces are going to be somewhat black. And then again, we're going to look towards the tail of the bees for these. Bombus fervidus, you remember in the middle of T6, usually has a little bit of yellow hair in the middle. You can't see it in this photo here. But you'll remember that Bombus flavidus is going to have the red at the very tip of the tail, which Bombus fervidus usually does not have, although it can. And then Bombus insularis on the right is remember it's going to have some break in the yellow banding near T4 or T5. And then it's also going to have that patch of yellow hairs right by its antennae, and that's what that arrow is pointing to there on the far right. So those are the T1 black bees. There's some of the other ones that could be confused for each other. Moving on to the T1 yellow bees. This is Bombus flavifrons versus Bombus centralis, which have nearly identical color patterns. There's a few things that we can look at to try to distinguish these two species from each other. Number one on the left, this is Bombus flavifrons. What we look for is that first arrow that you can see up here near the thorax. This is pointing to behind the wings. There is usually, almost always, some black hairs mixed in behind the wings in Bombus flavifrons. It doesn't quite form that V that we see in Bombus bifarius, but there are black hairs mixed in there. It doesn't go all the way through, but there are some. Comparatively, when we look in Bombus centralis over here, those are going to be all yellow hairs, or they don't go as deep. So again, things get really relative and challenging, but usually when we compare them, we see a lot more black hairs in Bombus flavifrons than we do in Bombus centralis. Likewise, you remember when we looked at the yellow versions of Bombus flavifrons, there were some black hairs intermixed at the front of T3 and the front of T4. That is also true for the males that have red on them. There should also be some black hairs mixed in on the sides of the abdomen for Bombus flavifrons, and there are not in Bombus centralis. I will say that I find differentiating these two species from each other maddeningly frustrating. They occur in the same region at similar elevations, and I have a hard time with this species group. I don't like it. I feel like I have to make uncomfortable decisions sometimes, and I think the older I get and the more experienced I get, I tend to put Bombus sp on these more than I'd like. So yeah, go ahead, Leif.
Just barge in for a second. I agree with Rich. This is a very hard comparison. The relative amount of black hair on the thorax for flavifrons it diminishes as you move southward, and so in the Sierra Nevadas, it can be especially difficult to distinguish the two. Whereas a little bit farther north in the Pacific Northwest, for example, you're generally going to see that mix of black and yellow hair on the thorax that Rich described. A second thing is that people will say that centralis is a lower elevation bee than flavifrons. I'm not sure that's true. Looking at the data, looking at where we're finding them lately, but that is a truism from Bumblebee world. So do what you will with it.
It could just, the higher elevation ones just could come from bad IDs from me. But no, I think you're right. I think I see this bee at higher elevation. And the females are easier to distinguish than the males. So, all right, looks like I need to keep moving here. So I'm going to, so these are the fuzzy T1 yellows that also have red on them. Bombus melanopygus on the left here, Bombus sylvicola on the right. You'll remember from the females that Bombus sylvicola is a very high elevation species, but Bombus melanopygus can also be found at fairly high elevation. So there's certainly overlap there. If you're at lower elevations, you're not going to find Bombus sylvicola, at least at our latitudes. So that's something you can use to distinguish these two species from each other. The other things that we'll look at are the thorax. So the front of the thorax, you remember in Bombus melanopygus, there we have that cloudiness. There's no cloudiness in Bombus sylvicola. I will say that this character is variable. Again, as Leif mentioned, the further south you go, the fewer black hairs there are intermixed or the less cloudiness there is there, and the more yellow hairs. Thankfully for Bombus melanopygus, the further south we go, we start to lose the red hairs as well. So this becomes the Edwardsi color form once we cross over from Oregon into California. So you wouldn't confuse these two species with each other, although sometimes Bombus clavicola also loses its red. So it can get confusing. So yeah, we're going to look at the front of the thorax for those species. Oh, also, I want to go back here. Also on T4, where the yellow is, usually there's some black mixed in there for Bombus melanopygus, and no black mixed in for Bombus cervicola, or less black mixed in, or if the black's mixed in, it's only in the center and not across it, as we usually see in Bombus melanopygus. So that's what I would use to distinguish those two species from each other. These are the less fuzzy yellow, red, yellow T1 yellow bees. Bombus huntii, just like the females, the males are nice and uniform. They almost always look just like this: yellow, red, yellow. Their T5 is also yellow on huntii males. We see similar color patterns with Bombus bifarius. So these two species are in the same subgenus. We can confuse them from each other. But usually in the center of T2, Bombus bifarius has yellow, and that is never true for Bombus huntii. So we can look at that section. It often comes down kind of like a rectangle. The other thing that we often look for is on the sides of the where the corbicula would be in the females. It's almost always red in Bombus bifarius, and it can be sort of pale colored in some of the other species, but it's really red or amber on Bombus bifarius. So those are good characters to look for as well. Again, these are yellow, black, yellow males. So Bombus, so the red here has been replaced with black. Bombus melanopygus in the left, Bombus bifarius on the right. Two things that we can look at to distinguish these two species from each other. Again, then the black notch behind the wings that we see in females persists in males. Sometimes it can be harder to see because the hairs are longer and they come up and cover it up. But if you were to push those aside, you'd find the black goes all the way to the back of the thorax there. And that is less pronounced in melanopygus. Likewise, sometimes, again, the further north, the more reliable this is. Sometimes there's some more black hairs mixed in at the front of the thorax for Bombus melanopygus, and something that you can use. That will be less true in Bombus bifarius, although still true. Bombus rufocinctus is its own beast. It can be really hard to ID. I think as the females, it can look like a lot of other species. It has big eyes, but the eyes are not super big. So it can be a really hard character to look at. So instead of pulling up instead of having one photo or two photos to show you, I just put a bunch of Bumblebee Watch observations here that I thought we would just pop over and take a look at, so that you can see what rufocinctus eyes look like in combination with some other characters. So you can see, if you just sort of open this up and look at it from here, you might start to see, well, this is a bee that maybe T1 is yellow, maybe it has big eyes. I'm not really sure. It's really hard to tell. This is a hard bee to be certain with. It also kind of has a red tail. So it has like all the features that you would expect in a bee. You could also think, oh, maybe this is a yellow teddy bear with some orangish on there. So, you could confuse this for almost any of the groupings that came to be. I do think when you look from the top here, that you can start to see a little bit of bulging on those eyes. They're starting to pop forward a little bit. It's not nevidenzis, it's not Morrisoni, but you can see it a little bit here. And if you look at it from this perspective, you can see it a little bit more. And I wish this zoom feature actually wasn't working so that I could show you this. But this space here on the right, you can see it's just the tiniest little cheek. It's the shortest cheek you can possibly imagine. And this is a character for rufocinctus that holds with the males and the females. You'll also notice, if you look from the top, just like kind of how boxy this species is. It's got short, even hair, and it's just kind of boxy all over. And that's a character that really holds true for Bombus Rufosinctus. I'll show you a couple more, and then we'll try to save a couple minutes for questions. So here, once again, from the top down, you can just see those little bit of bulging on the eyes. It's not hugely evident, but it is there. Again, it almost looks like a yellow, red, red, yellow, yellow male, so it can be quite confusing. But again, if we look from the top down, you can see how tiny that cheek is and that those eyes indeed are a little bit bulging. Is there anything that's really all that different? Yeah, here's just another example. You can see how boxy this species is. And again, you can see those eyes, how they're starting to pop out a little bit in front of the head. The rest of these are more or less the same. It's just trying to show you that this is a confusing bee, but cueing in on that cheek length and taking a picture of that cheek length, importantly, if you're doing surveys, is really important. And it'll help you see that those eyes are bigger than your typical sort of male bumblebee eyes. Just want to remind you that there are a ton of identification resources out there. Bumblebees in North America is kind of the authority right now. It has a key to males and females in the back and can be really helpful. There's some photos to help walk you through some of these characters. The Bumblebees of the Western United States book is great, but there's no male key in the back. There's only a female key, so it's limited in that regard. And then there's Robin Thorpe's guide for California, which has wonderful keys in the back of it. Bug Guide is a good option. Bumblebee Watch is an option, and iNaturalist are all other good places online that you can go for Bumblebee ID. Hopefully we've spurred your interest and reminded you that bumblebees will be flying in a few short months, at least in California, and then soon thereafter and moving up through the latitudes. I hope you've learned something. I hope we've entertained you a little bit, and thanks for coming.
All right. And so we don't have any questions registered. So if you have one, please go ahead and put it right in the Q&A. And we can take a few more minutes if necessary.
We were clear as mud, apparently.
So simple. Yeah, they got it all.
That's a good question, Dylan. How's it over already is something I want to know as well. It goes so fast.
All right, we do have a question. Will the slides be available? We plan to make this presentation available. We've recorded it. And so in about 1 to 2 weeks, it will appear on the Xerces Society's YouTube channel. So go to YouTube and search for Xerces Society and you'll find us. Yeah, it'll appear there in 1 to 2 weeks.
Leaf, is it a good idea for us to share the flowchart as a PDF? Could we make a PDF of that and maybe post it on the Atlas websites for folks?
Yeah. What if I could email the participants tonight when we post this on YouTube, and I'll include the flowchart, and I might also include a little Excel table that's a matrix of the characters, so you can kind of see bins the bees fit in. We've shown you that it's more than one bin in some cases. So the female BioV just asked, thanks, is that the same for the females presentation? Yes, that is already on the YouTube channel, so go have at it. Michelle Graydon asks, can you explain haplodiploidy in bumblebees in a really basic way? I'm going to try. Okay, so bumblebees, like a lot of other Hymenoptera and other insects they're related to, the sex determination is different from human beings, where we have an X and a Y chromosome, and if you have one of each, you're male, and if you have 2 X's, you're female, and there are some intersex conditions with multiple, right, and so on. In the bees, the way it works is there's a single locus with many different versions, not just X and Y, but you could say A through Z maybe. In fact, sometimes more than 40 different versions. If a bee is diploid, so it's a fertilized egg, and they have 2 different versions of the gene, that is a female. If it's diploid, so it has 2 sets of chromosomes, and the 2 versions of the gene are identical, that is going to turn out to be something that looks like a female bee, but is actually, it's going to turn out to look like a male bee, but it's actually a sterile male who cannot reproduce. It does live, it takes up ecological space, if you will, but it does not contribute to reproduction. Real males, healthy males, are haploid. So they are eggs laid by females that have not been fertilized. And the queen can actually choose whether to fertilize the eggs or not. So when she wants to make male offspring, she just lays the eggs that are just haploid. They're just her cells. So the males are 100% related to the mother in that case. And when she wants to make a worker or a new queen, it's a diploid egg. And so finally, just quickly, the big consequence here is that when genetic diversity is really low, populations are shrinking, the number of versions of the sex-determining gene get smaller, and eventually you have lots of diploid males in the population, and we call it an extinction vortex, where the rarer they get, the rarer they get.
That was pretty good, Lee. That was short, sweet. I don't know if it was sweet, but it was short. It was good.
Stephanie asked, do you know when the Atlas training for 2024 will start? In California, I'm not sure you're in California, but it will start in February probably, with a March 15th opening season date. But that will vary in other parts of the nation.
Yeah, if you're joining us from the Pacific Northwest, our Atlas as we know it is going to change. Our grid system will disappear. Folks can continue doing surveys and we'll continue identifying bees. But we're going to shift to more longer term monitoring. So this winter, we're going to be designing and developing a long-term monitoring protocol where we will be establishing what we hope will be permanent sites throughout the region, and then looking for volunteers to help us to do surveys throughout the summer. So like every 3 weeks at those sites. So keep your eyes peeled on the Pacific Northwest Bumble Bee Atlas. We're still wrapping up the last 6 years of our project and haven't shifted to looking to the future as much yet, but stay tuned, and we'll keep you updated as that program evolves. But there should be information out in the spring about it.
We have 2 more questions. Maybe we can quickly get through these. Dylan asked, is there something different about crotchy eye and rufous cinctus eyes? And yeah, crotchy eye eyes are much larger than rufous cinctus eyes, and you saw examples of both of those bee species in this talk. I wanted to quickly say something I forgot to say. I think this is fascinating. The reason these males have such large eyes, those that do, is it related to how they find females. And so those species that have the large eyes are sort of sit and wait males. They are territorial. They will mark a little territory with pheromones, and then they go sit in the middle of it at like the highest place, maybe a piece of grass or something. And they sit there waiting and looking for females. And you can fool them by throwing a little twig or stick into or pebble into their territory, and they will chase it. Other bumblebee species, the males find females differently. They probably are using pheromones, but they're finding them at the nest entrance or doing other things. So these large-eyed males are sort of cool ecologically to me.
Do you want to handle that last question, Julie?
It says, I've noticed that male bees might have longer colored hairs that appear to stick upright more than the females, and therefore look more banded above the integument. Yeah, the males can be a lot shaggier than the females, and so sometimes you see less of the integument. I'm thinking of the picture of Sylvicola that Rich showed a few minutes ago. It was just so bushy, you can hardly see anything underneath the hair. And females can often be quite short-haired, as you know, and then when they're working their abdomen back and forth to breathe, you will often see the black integument in between the segments. And so, for example, you saw some photos of Van Dykii, where Rich told you that they're entirely yellow on T3 and T4, but you could see some black color there. That was the integument, the skin. It was not the hair. So, so pay attention to whether you're seeing hair or not.
Yeah, and there are a fair number of male bees that have quite short hair as well. Nevidensis and morrisoni being 2 of them, where you can see quite a bit of the integument. And the male, yeah, everything else I think you covered. Good.
Anything else?
Looks like we covered it, Leaf. This was fun. I'm glad we did this. It feels like we finally gave gave males their due time in the sun.
Yeah.
And I'm glad it's recorded and we can get it out to folks so they can continue to watch it.
Yeah. And so thanks for coming and joining us. We're really excited to see you next spring in California if you're participating in that Bumble Bee Atlas, or elsewhere if you're participating, as Rich was talking about. So yeah, thanks and have a good rest of December.
See you next year.
All right. Bye.
Bye. Thanks.